Gene expression is the process that turns the static code in our DNA into the working molecules that make cells behave, mostly proteins, but also functional RNAs. It isn't a single on-off switch, it's a multi-layered choreography that makes sure the right products appear in the right cells, at the right times and in the right amounts. That choreography is carried out through a series of connected control points, from how DNA is packaged to how proteins are modified or discarded after they're made. At the top of the hierarchy is chromatin and epigenetic control. How DNA is wrapped around histones and folded into higher-order structures dictates whether the transcription machinery can reach a gene. Nucleosome positioning and compaction can hide promoters and enhancers or leave them exposed, and chemical tags on histones, acetylation, methylation, phosphorylation, and others, both change chromatin's physical state and recruit proteins that enforce activity or silence. DNA methylation, especially at CPG sites in vertebrates, acts as a more stable silencing mark and plays roles in imprinting and exon activation. ADP-dependent chromatin remodelers can slide or evict nucleosomes to reveal regulatory sequences, and non-coding RNAs, including long RNAs, often act as guides to bring chromatin-modifying complexes to particular loci, tuning the local epigenetic landscape. Transcriptional control is another major layer. The process of assembling RNA polymerase and the general transcription factors at a promoter is itself highly regulated, with core elements like TATA boxes and initiator sequences helping position the machinery. On top of that, gene-specific transcription factors bind enhancers, silencers, and promoters to recruit coactivators or co-repressors. This combinatorial binding is a key source of cell type specificity. Enhancers, even distant ones, can loop to promoters, and complexes such as mediator and cohesin help bridge these long-range interactions. Some transcription factors respond directly to extracellular signals, think steroid receptors, NF-kappa B, or AP1, so the program of gene expression can be rapidly reconfigured. Even after transcription starts, regulation continues. RNA polymerase II may pause near the promoter and only proceed into productive elongation when factors like PTEFB release it. What happens to the RNA matters just as much as how it was made. Primary transcripts are processed, a 5' cap is added, introns are spliced out and exons joined, and a 3' is cleaved and polyadenylated. These steps affect stability, export, and how efficiently the message is translated. Alternative splicing expands the repertoire of proteins a single gene can produce, guided by splicing factors that recognize enhancers or silencers within the pre-mRNA. RNAs are also chemically edited and modified. M6A methylation is a prominent example, which can change their fate in terms of stability, localization, and translation. Export to the cytoplasm is selective and mediated by export pathways and RNA-binding proteins, and once in the cytoplasm, mRNA lifetimes are tuned by sequences in the 3' untranslated region. AU-rich elements, microRNA binding sites, and protein binding motifs can all hasten or delay decay, allowing rapid shifts in protein output without altering transcription. Small and long non-coding RNAs add yet another dimension. MicroRNAs and siRNAs, through Argonaute-containing complexes, find complementary mRNAs and suppress their translation or trigger degradation, acting as potent post-transcriptional regulators. Long non-coding RNAs are versatile. They can scaffold protein complexes, act at chromatin to influence gene expression, or sponge up microRNAs, indirectly affecting mRNA targets. At the stage of translation, cells focus on initiation as the primary control point. The availability and activity of initiation factors, the presence of upstream open reading frames, internal ribosome entry sites, and structural features in the 5' UTR all influence whether an mRNA is translated and how efficiently. Global signals, nutrient status, growth factors, or stress feed into pathways like mTOR or the integrated stress response to adjust the translational machinery, enabling swift changes in protein synthesis independent of transcription. Even after proteins are made, their story isn't over. Post-translational modifications, phosphorylation, glycosylation, acetylation, lipidation, and ubiquitination change a protein's activity, interactions, localization, and stability. Cells also control protein abundance and quality through targeted degradation. The ubiquitin proteasome system and autophagy remove unwanted or damaged proteins, while chaperones and ER-associated degradation pathways help fold proteins correctly or target misfolded ones for disposal. All these mechanisms are woven into networks full of feedback and feed-forward loops. Transcription factors regulate other regulators and signaling components, and often their own expression, creating dynamic regulatory circuits. Spatial and temporal specificity, local chromatin environments, cell type-specific factor expression, and even localized translation adds contextual nuance so that gene expression programs are tuned to developmental stage, tissue type, and environmental cues. These layers of control have clear biological consequences. The lac operon is a classic, simple illustration of how a repressor and an inducer can switch metabolic genes on and off in bacteria. In multicellular organisms, development depends on enhancers, combinatorial transcription factor codes, and epigenetic memory to establish and stabilize cell identity. When these systems break down, the results can be severe. Cancers often involve mutations in transcription factors, chromatin regulators, or signaling pathways, while neurological and other disorders can arise from splicing defects or disrupted RNA metabolism. In short, gene expression is a dynamic, multi-layered network that integrates genetic information with cellular and environmental signals to produce precise, context-appropriate outcomes, an elegant balance of control points that together make life both robust and responsive.